faster than the regular single-stranded RNA virus. And if you look at particularly a gene that's called the S gene, I'm going to come back to it again and again. And this S gene will code for a protein that is responsible for binding to the host receptor. And this substitution overall is on par with the single-stranded RNA virus, but the RNA binding site, it almost has 10 times the faster evolutionary rate. And this is really essential for the virus to really find in a host and then find a potential way to replicate itself instead of just getting killed by the host immune system. And then recombination is another way that the virus would evolve. And recombination, essentially, you have viruses, multiple viruses, maybe infecting a host, and it simply switched the template because it doesn't actually go from the beginning to the end. It would actually go from little islands, try to use a template to replicate itself. But sometimes it uses one template and sometimes it uses another, and therefore the breakpoints seem to be random. So you have these two mechanisms that essentially create the intra-species variability, and I'm going to show some of that later, and also the interspecies host job. And people have done the phylogenetic analysis of the COVID-19 coronavirus, and we call it 2019 nCoV, or the more standardized name is called the SARS-nCoV-2. Right? So you can see in publication, because it's so new, people have published in different names. And this is a bunch of sequences that people collected from all over the world. And these are the new viruses. And then there's nothing like that in this clade. And if you go to the closest one, is the beta coronavirus that were derived from that. And then the SARS is even farther away from the evolutionary distance. So in this tree is a phylogenetic tree. The closer the species are, then I think the closer that they are supposed to be nested to each other. And then farther away are the MERS and the Asian flu, and so on. So by now you should get a sense that this is indeed a new beta coronavirus. But what about the genome structure? And it turned out that all the coronaviruses have more or less the same genome structure. So the genome will vary between 30,000 to 32,000 depending on the strain and the species. And in this particular figure, they show the SARS and the MERS. And you can see that they almost are identical. And there's actually very, very long open reading frame that's called the ORF1A and 1B that encode a couple of essential proteins. For example, this RDRP encodes for an enzyme called the RNA-dependent RNA polymerase that is very essential for the virus to use the host system to do replication. And then the helicase is also responsible for viral replication. It's very long open reading frame that encodes multiple genes. And on the other side, you will see that there are proteins, especially the structural proteins. We just call it SENN. So these S is the spike proteins, and I'll come back again and again. It's really long. It formed this a crown-like structure, and it's actually responsible for binding. And then the E is the amyloid proteins that actually cross membrane. And then there are membrane proteins and also there's other proteins that are pretty much essential for packaging the genome. Now for the new SARS, the new coronavirus, and this is the structure. Throughout this ORF1AB, you can see that it encodes up to 16 genes in a very long continuous region. And then the spike protein has a receptor binding domain, and then there are actually other EM and some other proteins that are genes that are encoded. When it doesn't have a special name, and usually it's not that essential for the virulence. How does this new coronavirus genome compare against other known beta coronavirus or the SARS? Right? Now, I want you to look at this chart. So this is the entire length of the genome, but then shortened and broken up into these proteins, right? Open reading frame 1AB, and 1B actually has this RNA-dependent RNA polymerase, and this S is a spike. Right? So you can see this red color is a SARS that are actually quite distant from this, which are the bad beta coronavirus. Except for a few key regions, such as the S protein, there are parts of the regions where the SARS seems to be more similar. But overall, if you see the similarity of the SARS genome to this genome is about 79%, and the beta coronavirus is about 87%. Obviously, none of these three genomes that are being compared can be the real ancestor of this virus outbreak. Now, people have further used the bioinformatics method and to really look at the protein level sequence alignment. So what is shown here is the actually multiple sequence alignment. Is one of the key methods that's used in bioinformatics. In fact, I'm going to do a shameless advertisement. If you're interested in how to do this, you should take the Info 701, Introduction to Bioinformatics. And I think we teach that. So if you look at the red ones, these are the SARS. These are the SARS residues, amino acids, for the S1 domain of the spike protein and S2 domain of the spike protein. In this representation, only the key residues are shown. And then when it has a dot, it means it agrees with the template, which is the first line. Now you look at this, the new coronavirus. The new coronavirus, actually for this spike protein, the key residues, surprisingly, it is very similar to SARS than the actual close beta coronavirus that at the genome level is similar to. So it really says two things. One is virus classification is a very specialized task only the expert probably should do. If you just do it at the genome level, you may actually misclassify this. You really have to find the right sequence to do it. And the article really talked about some interesting aspects. They did point out the dilemma. So here is a new coronavirus, and it's far away from the SARS, and then it's closer to the beta coronavirus. So from the whole genome phylogenetic tree level, it should be a new species by itself. But this Lansley paper also were puzzled. Should we actually classify this as a SARS-like coronavirus, in fact with R? Because for this particular protein, RDB binding domain of the S protein, it actually is classified, is actually closer to this. And there's there's more evidence. They look at the SARS binding to the receptor of human called ACE2 protein. They look at the key residues and the structural fold of the protein. And they found out that they suspected that this new coronavirus should also bind ACE2 in human. However, all of the contact residues seem to be different. So what is going on? Right? So a lot of people are starting, after reading this paper, starting to have the conspiracy theory to say, wow, maybe someone just engineered this from a SARS and just put it in here. Right? So that's certainly one of the disputes. And then there's even some Indian authors published in a preprint and try to argue that possibly this should be an interpretation, but after the expert checked on their paper, there was quite of a nonsense. So we're not completely ruling out all the hypotheses. As I said earlier, I'm not going to actually help you if you have that question, but at least scientifically, you can see that there are closer sequences that can be aligned. So another paper published in Nature found out another genome that's called RATG13. And RATG13 is a beta coronavirus that were previously not deposited in NCBI's database. Look at the similarity of the new coronavirus with RATG13. It's far exceed all of the other ones. And when you do the classification, the new coronavirus found in the patients also are clustered together with the rat RATG13. So it seems like the problem has been